So I'm going to start right away. Some stuff I'm just going to explain. So the parts of, external genitalia, scrotum and penis, right? Now, what inside the scrotum that I want you to kind of appreciate? There are two muscles, okay? Dartus and cremaster. So these two muscles ascend and descend testes. Okay? Now, what regulates ascend and descend? What is the main function of testes? Maturation of sperm. Does that make sense? Sperm has to mature at very precise temperatures. Am I clear? So, listen, just look. When it's too hot outside, to cool down, testes descend. Does that make sense? When it's cold, they ascend back inside the body. And this process, it's not like yes, no, up, down. It's a regulated process to maintain testes at a certain temperature. As far as I remember, it's about 34 degrees Celsius. So it's not full 37, it's not too cold. Is that clear? That makes sense? FYI, some people try temperature change as a birth control method on the male side. I don't know how well it works. I would not rely on that. Okay, people would, put testicles into hot water, tolerably hot water, expecting it to inactivate the sperm. I have serious doubts about that, considering the amount of sperm cells that are there. You're missing half of them, and that's still a lot. Okay, now, anatomically. Let's talk about the path for sperm from testes all the way to the urethral orifice, okay? So we start at seminiferous tubules. Niferous tubules, okay? So we start there. So this is where sperm is generated, sperm cells. Then we go to straight tubules. Then we go to a little network called retitestes. And then it goes into the I hope I spell it right. I'm not sure. Okay, epididymis. And then it's very, very long and convoluted tube, okay? I have no idea how long it is in humans, but I worked with male mice, and in male mice, the epididymis is like good 5 times longer than the mouse itself. It's pretty long. Vas or ductus deferens. Ever heard about, cutting the tubes or ligating the tubes or tying the tubes, the birth control in males? This one. At some point it was just snipped. Right now, as far as I know, some people prefer to tie them. So in case a male wants, to have babies, there's a chance that they will retain reproductive function, and can be untied. Now, here's the important part. So, at the ejaculatory duct, there's like a couple of glands I'm going to add here. Seminal gland gets right around ejaculatory duct. That makes sense? An ejaculatory duct merges into the urethra. Okay? And there is a basically at the merge of ejaculatory duct you have a prostate gland, which is important, and then vulvourethral gland here. Okay, so let's go over this process of sperm travel step by step. So just follow me on that. I'm going to quickly review it. Look, sperm cells generated into the seminiferous tubules, okay? They mature there, they move to straight tubules, mature there, move to retitestis, continue maturation, move to epididymis. They can sit in epididymis up to 2 months, still maturing. Then, during ejaculation, they move through vas deferens, follow me here, into the ejaculatory duct, okay? And then ejaculatory duct merges into the urethra. Kind of interesting thing, there's a little sphincter that separates urethra from the urinary bladder. When a man ejaculates, when there's the spasm that moves semen through the ejaculatory duct and urethra, does that make sense? The sphincter between urethra and the urinary bladder closes. So sperm, the semen, doesn't get into the urinary bladder. Does that make sense? Basically, I can put it this way. So you've got urinary bladder right here. So when ejaculation happens, look, this little sphincter here closes. So semen can only go one way. Now, what's the function of these glands? Seminal gland, prostate gland, their secretions, they do secrete, okay, they're exocrine glands, they secrete the liquid component of called sperm, okay? So sperm cells are coming from here, and the liquid component that contains things like fructose, energy source for sperm cells, okay? Coagulation factors and plasmin. You need sperm. It's actually very interesting. Look, when sperm gets into the vagina, it first coagulates to prevent it from leaking out. Does that make sense? But then plasmin breaks down those coagulation factors, so the sperm cells can actually be released and move towards, secondary oocyte. Am I clear? It's slightly basic, semen is slightly basic, because urethra in males, check this out, urethra in males also provides the path for urine, right? And urine is slightly acidic, so it neutralizes acid. And vaginal pH is slightly acidic. So this base basically, provides for neutralization. Okay, makes sense? Now, in terms of the bulbourethral glands, they produce lubricant for the glans penis. Okay, a lot of anatomy I'm not touching here, right? So, basically the outer surface of the penis is covered in mucosal kind of secretion from bulbourethral gland. Okay? That makes sense. The mechanism of response, I think we talked about it in AB1. I will remind you briefly. So you have two systems involved, okay? You have parasympathetic, and parasympathetic system results in the following. So you have, ACH in vasculature, okay, which increases the release of nitric oxide, I'm talking about penis now, okay? Does that make sense? So, anatomically, listen, anatomically, penis looks like a sponge if you cut it open. Does that make sense? It's three bodies, two that are called corpora cavernosa, cavernous bodies, and one at the bottom which is called corpus spongiosum. Okay? So cavernous or spongy. There are no muscles like in the traditional sense. I mean, obviously there's smooth muscle around the blood vessels, but penis contains a lot of blood vessels. Does that make sense? So parasympathetic stimulation leads to increased NO, vasodilation, and engorgement of penis with blood, basically, and erection. Okay, so the famous erectile dysfunction drugs, such as Viagra and Cialis. Well, first one was Viagra from Pfizer. It was originally the drug that was designed to fight high blood pressure through vasodilation. That makes sense? And the legend, the apocryphal legend has it that the CEO of Pfizer was talking to his personal trainer in the gym, sharing that they have a phenomenal drug to treat high blood pressure, but it has a weird side effect. And the personal trainer looked at him and said, "You're an idiot? It's going to be a gold mine." And they remarketed it for erectile dysfunction. It still works, it still reduces the blood pressure. This is one of the big side effects, that in people who have normal blood pressure, like in males with normal blood pressure, one of the possible side effects is the dangerous drop in blood pressure if they take Viagra or Cialis. Does that make sense? Now, what is sympathetic system doing here? So sympathetic system smooth muscle contraction and ejaculation. Are we clear? That part. Okay. All right. Now, I want to say now. I'm going to say, you know what, let's move on to, I'm just trying to process what I need to touch upon. So we've got the path, we've got the release of glandular secretions. Okay, let's do the hormonal regulation. And we talked about it before, it was a part of a Unit 1, so it's more of a reminder, some kind of deeper dive into this, okay? So, in terms of the hormones, it all starts in hypothalamus, which produces gonadotropin-releasing hormone. Okay, we good? Gonadotropin-releasing hormone stimulates anterior pituitary gland, which produces two gonadotropins. One is luteinizing hormone, and the other one FSH, follicle-stimulating hormone. Okay. So luteinizing hormone, both of them act on testes. So luteinizing hormone acts on testis specifically, and that's important, listen, it's called Leydig cells, or sometimes called interstitial cells. FSH also acts on testes, but this time it's Sertoli cells. Okay, you with me? Sertoli, S-E-R-T-O-L-I. Are we clear? So Leydig cells produce testosterone. And Sertoli cells produce androgen-binding peptide. Does that make sense? And it turns out that both of them, androgen-binding peptide and testosterone, look at this, are critical for sperm maturation. We're good? Now, here's an interesting story. First of all, testosterone is inhibitory. So it provides a negative feedback here. Now remember our previous conversations about negative feedback, how it is a teeter-totter? You have to appreciate this. Look, the fact that testosterone inhibits both hypothalamic release of GnRH and release of gonadotropins from anterior pituitary doesn't mean that it stops them totally. It just means that it kind of keeps it in check. You see what I'm saying? So you have a fine balance between testosterone, gonadotropins, GnRH, which means a male generates sperm all the time, but the rate of sperm generation is very, very tightly controlled. You see what I'm saying? Because if there would be no negative feedback, this thing would blow out of proportion. And quite literally, like the amount of cells, it would blow the testis up just by the sheer volume. Okay, so it doesn't happen. Another interesting thing here. So sperm count, not concentration, but physical, like how much of it increases, right? So sperm fills, I got it out of here, but sperm fills seminiferous tubules when it generates and then goes into the straight tubules, retitestis, epididymis. It accumulates and accumulates and accumulates. Now, here's an interesting thing, still kind of out of control, isn't it? So, a more sperm, I'm going to make like a big bracket here. It will produce the hormone called inhibin. The higher the sperm counts, the more inhibin is produced. Does that make sense? Now, which process reduces the sperm counts? Ejaculation. You see what I'm saying? So when a male ejaculates, the inhibin-induced inhibition goes down, levels of these hormones go up, more sperm produced, right? More sperm, more inhibin, so sperm production slows down. Does that make sense? When another, each ejaculation reduces the number of sperm cells, which reduces the inhibin-induced inhibition, so to say. Got it? The numbers in males are absolutely whopping. I do not remember. Like, we're talking, like every ejaculation, we're talking millions, if not tens of millions. It's enormous. It's such a waste of resources, and it kind of suggests that nature is not about optimization. If it works, it works, Now, something I wanted to mention here. Oh, yeah. Testosterone. Let's say a few words about this hormone. Testosterone in males, levels of testosterone spike during puberty, right? So basically that explains why males become more or less like functionally fertile around puberty. Now, what else happens to a boy during puberty? Growth spurt. Does that make sense? So that's testosterone. Okay, see that? So growth spurt. Skin becomes oily, bones become thicker, the muscles start to grow. That makes sense? Sounds really attractive as the performance enhancement drug. Are you following me here? Now let's take a look. What's going to happen if an athlete takes testosterone as a performance enhancement drug? So if you take testosterone, what happens to, specifically, levels of gonadotropins? They decrease. Are you following me? Now, gonadotropins stimulate one and only organ, testis, right? And if a hormone doesn't stimulate the target organ, what happens to the target organ? It deteriorates, it gets smaller. So balls literally shrink, like literally shrink. And this shrinking is basically reversible. So if someone takes, there are many more testosterone associated problems, obviously. But for instance, one of the side effects are basically acne. I'm not going to go into microbiology of it. But so two sort of big consequences: shrinking testicles. If somebody like uncontrollably starts just popping testosterone all the time, right? The lack of stimulation of testicular tissue results in testicles shrinking, and that results in infertility in the long-term perspective. That makes sense? Okay. Now, last few things about males, and that's going to be very simple. We used to go through the entire meiosis, and it was such a pain in the ass. So what I want you, like the actual processes, like Christian, go over, what's that, when chromatids are distributed unevenly, that kind of stuff. Like, no, not anymore, okay? So what I want you to understand, it all starts with cells that are called spermatogonia, okay? So these are proper diploid cells with 46 chromosomes, 23 pairs. Does that make sense? Now, spermatogonia, fundamentally, please understand, that's fundamentally. So they undergo several steps. One step, they go through mitosis, okay, which produces two diploid cells. One diploid cell kind of gets stored for the future. Are you with me? Another diploid cell undergoes two meiotic divisions. So meiosis 1, look at it, generates two haploid cells. And then meiosis 2 generates ends up with 4 haploid cells. And this happens like all the time. Does that make sense? So it goes on and on and on and on. So I would like to tie these things down. So look what happens in a healthy male. In a healthy male, I'm going to skip first steps. Testosterone that's produced by Leydig cells and androgen-binding peptide that is produced by Sertoli cells, they guide the development of spermatogonia into the spermatozoa. This development involves 1 mitosis and 2 meiotic divisions. See that? First meiosis gives you 2 haploid, meaning only one set of 23 chromosomes. And the second meiotic division gives you 4 haploid. Are you following me? So the whole development of spermatozoa goes from 1 diploid to 4 haploids. We good? So kind of harkens back. If we go back to this, what is the function of Leydig cells? Testosterone. What is the function of Sertoli cells? Sertoli cells essentially guide the development. Does that make sense? They move it forward. They kind of, and they also protect sperm cells because they can be immunologically different from the rest of the body. They form a barrier between the blood and the sperm. Okay. Good? Clear? I think I covered what I need to cover here.